Welcome. We're so excited to share some information about our imaging work here at CBTN today. And as we get started, our goal today is really to make sure that this can be an interactive conversation. So I put into the chat, I realized that I was only sending it to other panelists, and that's why no one responded. There we go. So if you could put into the chat who you are and your organization and where you are in the world, that would be wonderful. Excellent. Thanks, Al Musella, who's the only person who responded in the chat. Thanks, Kim. Nice to see everybody. Excellent. So I'm going to be, both Ariana and I are going to ask for your participation today. So just to make sure that this is interactive, it's one of the things that we've been talking about. How do we work to inform you of all of the great things going on here in a way that you can interact with it and understand it, and to make it as meaningful as possible for you. So with that, I think I see a lot of you on this call today are folks who know quite a bit about CBTN. So I'm not going to do a full overview of who we are and what we do, but I do want to just reiterate that, as many of you know, CBTN enrolls subjects at the time of their diagnosis and then collects clinical data throughout their tumor journey. And during that time, all of those kids are getting images and scans. And Ariana is going to talk about the power of those scans and what we can learn from them and how it can inform treatment down the line. So I'm so excited to introduce Dr. Ariana Familiar, and you can who's here at the Translational Imaging Unit within the Center for Data-Driven Discovery in Biomedicine. And I am just really excited about all of her work. I know that many of you have heard me sort of wax poetically about her and the work that she's doing, but I'm excited for her to share all of this with you today. And please don't hesitate to ask questions. I'm going to be monitoring the chat throughout, and as you have questions, as you have feedback or ideas, please don't hesitate to ask them as they come up. So with that, I'm going to hand it over to Ariana. Join me in welcoming her today. And again, questions, comments, ideas, put them in the chat.
All right, awesome. Thank you so much for having me. I'm really excited to be here and to talk and tell you about some of our recent work using CBTN imaging data, ultimately with the goal of transforming patient care for our pediatric brain tumor patients. And so I'm going to start with kind of a poll to the audience here. So why do folks think that we even take MRIs of our pediatric brain tumor patients at all? So during clinical care, patients will go in for several MRIs. Why do folks think that that is relevant and something that should be done? Or why does it happen? I can't, oh, I couldn't see the chat.
She said to understand the growth of the tumor.
Exactly, yep.
To evaluate if treatment is working.
Yep.
I know that more of you have ideas about this. Disease monitoring.
Yeah, totally. You are all correct. Hit the nail on the head. So some of the clinical implications of these MRIs, they're used for detecting the tumor at baseline. Yes, to get more details, to narrow to potential types of cancer. So when that patient comes in, what type of cancer do they have? How does that inform their treatment plan? Additionally, like several people said, to monitor for changes in the tumor size over time. So to see whether the tumor is increasing in size or progressing, whether it's responding to treatment. or whether the treatment comes back later on and recurs. And to really illustrate what that looks like for a specific patient, I'm going to talk about an actual CBTN patient who was 13 years old at her first diagnosis. For the purpose of this talk, I'm going to refer to her as Jane, but please do note that that's not her real name. So Jane came in to the clinic. She had morning headaches, and additionally, she had nausea and vomiting. And so her doctor sent her in for her first MRI. And in that MRI, we saw, or the radiologist team saw that she did indeed have a tumor located in the part of the brain called the posterior fossa. So here I'm just showing you an MR image. This is kind of the side of her head looking in. Here's the nose and the back of the head for reference. And this kind of spherical, kind of brighter part of the image is a tumor. This is the brainstem, and this is the brain region called the cerebellum. So in this MRI, it was determined that Jane indeed had a brain tumor, and the radiologist, based on where the tumor was located and the way that it looked on the image, made their best guess and said, okay, this could be either an ependymoma or a type of astrocytoma. Her clinical care team then determined that she would go in the next day for surgery. The surgeon then took out most of the tumor, but left a little bit of residual tumor, given that, let's go forward. Given that this specific type of tumor is not as aggressive as others. So they left a little bit of tumor in her brain. And this is the remaining of Jane's treatment journey over time. So she, after her first surgery, had several MRIs to examine how she was responded to that surgery. After both 6 months, she had 3 MRIs, and then within 6 months to 1 year, she had a couple more MRIs. And then at that point, a little bit after, it was determined then by the radiologist team that that residual tumor had indeed grown and was growing just very slowly over time. So then Jane started her first chemotherapy trial. She was on that for a year. Again, she went in for routine regular MRIs to see whether she was responding to the treatment, whether that tumor was growing or shrinking. After that, after 8.5 months of being on the chemotherapy treatment, the radiologist team determined that the tumor was indeed still growing, just slowly. And then she received more MRIs. She went off chemo. She then eventually progressed to a point where she had to have a second surgery. And then after 3 years since her initial diagnosis, she started a second round of a different type of chemo. And her most recent MRI it's now been over 4.5 years since that first diagnosis, that first MRI that she came in. So that's Jane's treatment journey so far. And I just, again, want to poll the audience and see what people think about this timeline. So what might you say could have been improved, or what might be unsettling about this picture, particularly thinking about Jane undergoing different types of treatments, undergoing her and her family waiting for knowing from the clinical care team whether she was indeed responding to treatment or not. So I'm going to wait for an answer in the chat.
Amicella said should show volume of tumor at each point.
Yeah, exactly. It's wonderful. I feel like you know where I'm going with this. Yeah.
Better disease monitoring and diagnostics from Misha.
Yep, exactly. Speed up response to treatment.
Starting treatment right after surgery unless it was not recommended. Yep. It's almost like you guys have thought about these.
Not coupled with other data. Beautiful.
Not coupled with other data. Insider.
Yeah, right.
Yeah, this is great, guys.
So exactly. So why did Jane need so many MRIs before determining that her tumor was growing? Why did she have to be on that ineffective chemotherapy treatment for so long before the radiologist could determine that her tumor was still growing? Especially given that this chemotherapy treatment has significant side effects, has known long-term effects on her quality of life. And so to kind of dive into what actually happens at each of these time points from the radiologist perspective. So here I'm showing you again another MR, but this time from the top down. These are the eyes, the nose, back of the head is here. And this is the tumor, again, in the cerebellum area. So in that like kind of back part of the brain. And so typically what a radiologist does is they'll sit at their workstation, they'll look at this type of image and other types of images from a single exam. So one time when the patient went in to the MR scanner and several images were taken of their head. And the radiologist will then write out a report, so a descriptive text report of what they see in the image. So what is abnormal to them? What are some of the features of the tumor, the image properties, like, okay, I can see it's kind of bright here and dark over here. And then importantly, to measure tumor size and to compare that across time, radiologists take two-dimensional measurements of a tumor. So for a given radiologist, they might come in and they say, okay, this looks like the widest part of the tumor to me. So I'm going to draw a line there. And that's actually what they do in their software. They draw a line from one side of the tumor to the other. Then they'll say, okay, this is the second dimension, I think. It's perpendicular to the first line, and it kind of captures the max width of the tumor there. And so that's my, in their report, they'll say, okay, here is, let's say 10 centimeter wide by 5 centimeter wide or something, just made up for this example. And they'll write that into their report. And then that will be done across time. So each time the patient comes in, the radiologist will do this. They'll report these results to the clinical care team. But there's known issues with this approach. So that's the measurement one radiologist would make, right? But then a second person could come in and say, okay, well, I think that the widest part of the tumor is here. And I'm only going to focus on the bright parts because sometimes that can be more important for understanding response to treatment and assessing response. So a second radiologist could easily come in and make a very different judgment of the size of the tumor based on these 2D measurements. A third radiologist might come in and say, oh, well, I see this part over here, this dark part, that's a cyst. So I'm going to include that as part of my main tumor measurement. And these are my max diameters that I'm going to make. Again, a very different size comes out from those judgments. And a fourth could even come in and make a different measurement using these 2D. So this is a known issue with the way that radiologists report results from these exams. But this is actually how it's done in the clinic. And so clearly it's not consistent across radiologists. Even within a radiologist, there can be variability. And importantly, this results in different estimates of size. So thinking about a small residual tumor, sorry, I just saw pop up in the chat. Doesn't it also depend on the number of slices taken during that MRI? Certainly, yes. There's significant variability. That's like a wonderful point. There is significant variability in the types of images taken and how those are taken. So you can think about one camera, for example, takes a picture at really high resolution and another takes a picture at really low resolution and becomes blurry, or the number of slices, right? So the number of actual images taken at different levels throughout the brain can differ, both across scanners within a hospital and importantly also across scanners across hospitals, which causes major variability. And so these estimates of how big the tumor is at one time point and how that compares across time are not accurate and not consistent. Now, for pediatric cases specifically as well, compared to adults, pediatric brain tumors can have a lot of different types of subregions. So it doesn't really matter for the purpose of this talk what these subregions are. The point is that across different types of cancers, and even within some cancer types, the appearance of these tumors in our pediatric patients are varied. So this can, again, further complicate what parts of the tumor radiologists include in their measurements, what 2D parts of the tumor they use. And this, again, causes significant variability in estimates of how big a tumor is at a given time point and how that changes across time. So bringing this back to Jane's example, so I took here, these are actual MRIs from her a few time points during her first chemotherapy treatment. So this kind of brighter circular part right here is her residual tumor. And so you can kind of see, okay, there's, maybe not that much visual size difference between these first two time points. It kind of changes some shape here, but hard to determine whether it's bigger or not. But then finally, 8.5 months later, based on this MRI, the radiologist determined that that it had indeed grown in time. From one of the actual reports from these time exams that were taken, the radiologist wrote out, the residual tumor is very irregular in shape, making precise reproducible measurements difficult. So clearly this is a known issue, but this remains the standard for our patients to date. And it's just suboptimal. So just to reiterate what I've been talking about, tumors are frequently asymmetric, especially for where they show up in our pediatric patients in the brain. They can be really complex in their appearance on an MRI, and treatment like chemo and radiation can cause changes in MRIs that further complicate the assessment of main tumor regions. As a result, there's a lot of variability in how these measurements are done.
There's a couple questions.
Yeah.
Like why not use other sorts of other things, other tools like PET scans that measure?
Almy Stella has seen my slides, I think.
Yeah,
Yes, that
is very informed.
Yeah, that's awesome. Yes, why not use an automated program? That's exactly where we're going.
We're getting there, Al.
Yeah. And why not do a metabolic PET scan to see what's active? Yes, exactly. I love it. Don't say sorry. Why not do a metabolic PET scan? Exactly. So also an active area of research. So PET scans have shown particular efficacy for some adult gliomas, and there's some work being done to translate those to pediatrics, but they are a little bit more invasive. So our pediatric patients would have to get an injection of a tracer for PET scans to work, but that is an active area of research to see whether PET can be used to do better than this. All right, awesome. So, right, we need better methods of diagnosing and measuring brain tumors, characterizing them across time. We need them to be more accurate and to do so at the level of the patient. And so this is the type of problem that motivates our work with the MRI data, and particularly with the CBTN MRI data. So can we provide an accurate diagnosis based on that MRI at day 0? Instead of saying, it's ependymoma or astrocytoma, could we have said, no, Jane is a low-grade glioma, and this is how we're going to therefore treat her? Could we have more accurate tumor measurements so that Jane and her family did not have to wait years to see whether her tumor was growing or not? Could we have predicted that she was in the subset of low-grade glioma patients that was a little bit more likely to progress? Could we determine those patients who are at a higher risk of recurring compared to others? Could we determine progression sooner? Could we have a better method of imaging to guide surgical planning so that that surgeon in that first surgery knew to just go a little bit more aggressive and potentially reduce the likelihood of her residual tumor progressing? And can we determine molecular mutations based on imaging alone? So again, to help guide the specific treatment plan for each patient uniquely. Let's see, difference in head tilt, yes, will change. There's a lot of variability in these MRIs being taken. Are we not at the stage of 3D reconstruction to get a better view and quantification of it? Again, getting there, Kim work, getting there. Just give me a couple slides. Okay, so we are the Translational Imaging Research Unit within the Center for Data-Driven Discovery and Biomedicine at CHOP. We at the center have several multi-interdisciplinary teams, and we are all kind of focused on bringing in data, annotating that data, making it usable at large scales for pediatric brain tumor patients, and ultimately using that data to drive discovery, to drive cutting edge methodologies that can ultimately be have impact in a clinical context. Within the imaging unit, we have several ongoing initiatives focused on building out large-scale data sets of MRIs for our CBTN patients. We build out software tools and processing workflows. We do predictive research or predictive analytics in a research context using AI, which I'm also going to get into. And we also have several other ongoing initiatives to, again, build out methods and models and algorithms that can be impactful in patient care. We are a multidisciplinary team. There are radiologists, brain cancer researchers. We have neurosurgery residents who come and do research projects with us. We have clinical research coordinators who are patient-facing, and we also have data engineers and analysts and data scientists. So at a high level, AI now is pervasive, it's a hot topic, and it's kind of grown into a lot of different fields and domains. So AI has had a lot of success. If you sit back and think about it, things like advertising with targeted ads when you're scrolling through Instagram, right? Somehow Instagram knows exactly what ad to show you based on your past history and the past history of other users. Face recognition uses imaging data to, every time you open your iPhone with Face ID or use face recognition for some sort of security measure, AI there has learned based on tons of other images of faces, essentially. Self-driving cars becoming a reality. Again, using a type of imaging data to perform this really sophisticated task of navigating around roads. And the success of AI in these ways really depended on the availability of a ton of data. So there's just really like infinite amount of data in a lot of these contexts. And not only a lot of data, but good data. So again, the fact that advertising has had success is because they have tons of history and data sets on people's like search behaviors and how they click through websites and social media. The same, unfortunately, is not true in healthcare. AI has not had as much rapid success, primarily because I would argue there just isn't enough data available. Healthcare and like the 2D measurement type of issue in patient care is ripe for using AI, right? AI has shown success in similar tasks in other domains. So why is it not being used? Why is it not implemented to date? It hasn't been, it's because the AI models don't do well enough to be used reliably and successfully. And so this is something that really is the underlying driver of a lot of our work. It's to really use the power of AI and predictive analytics with real world data from our patients, from our CBTN data set, and use that to build real algorithms that can ultimately be translated into clinical decision making. And we do that through, and again, I'll touch on these throughout the talk, but we create large scale data sets. We prepare those data sets for use. We integrate it with other available data from CBTN, like the clinical and molecular data. We develop algorithms with those MRIs. And then ultimately, once those algorithms perform well, we can implement them as software tools so that they can ultimately be used in the clinic. So really, this is kind of the overarching goal. It's to develop better methods of characterizing disease and tailoring patient-specific treatments, which requires a lot of data, a lot of strong model development, and really this end-to-end ecosystem where research can inform clinical care and vice versa. This requires large-scale data sets, which hasn't been done to date in adult or pediatric brain cancers. And it requires the use of these MRIs with AI to inform clinical decision-making. All right, so let's get into what some people have been eagerly waiting for, I think. So how have we used CBTN MRIs, particularly with AI, to transform patient care. So I'm going to bring it back to this measurement example of tumor size. So like I said before, the standard to date is to make these 2D measurements for radiologists. Some of you have already asked, why don't we just automatically extract tumor measurements? And particularly, can we do so with AI? Ideally, what could happen is we would have a 3D measurement of a tumor and a way to model the tumor, because we know it's a 3D object in the brain. It's not a 2D object. It is this 3D, often asymmetrical mass. So can we reliably delineate the tumor from other parts of the brain? And can we use that to guide measurements? This can't really be done manually, at least in the clinic, you might guess, because it takes a really long time. So just sitting there and manually drawing out this, like you do in Microsoft Paint or something, it requires a long time to really delineate the tumor accurately. And that feature, that drawing capability, it's not available in all radiology software, although more modern ones do have it. And it's still prone to human variability. So it still requires consistency across radiologists to draw the same regions as exactly the same, which is not, does not happen. All right, so this is a initiative that's led by Dr. Anahita Fatih-Kazruni, who is a faculty instructor in the Department of Neurosurgery at CHOP, as well as Penn Medicine. So this project essentially looks at using AI and uses AI to perform this task automatically. So what we did is we had our imaging team manually draw out those 3D segmentations for 244 CBTN subjects. That number has grown now. But initially, for the first iteration, we took those 3D segmentations that were manually drawn and essentially fed it into an AI model. And what that model then could do was learn essentially how to do that 3D segmentation based on the labels that it saw and the images that it was given. Now what we can do is we can take that AI model that's learned these patterns and essentially feed it new MRIs from new patients and automatically generate these 3D segmentations. Importantly, we had to do this in pediatrics because, as apart from adults, because pediatric cases look different. The tumors look quite different from adult cases. And so all of these subregions had to be delineated separately. But now we have an algorithm that can work very well on pre-treatment pediatric brain MRIs. This is just an example of the model's performance. So here's a manually drawn 3D segmentation from our team, and here's the model prediction. And you can see the colors, right, and the regions, the boundaries overlap significantly. Okay, so now that we have these 3D measurements we can calculate tumor size, and that has widespread clinical applications, right? So we can use that size judgment to both detect and potentially diagnose the patient at day 0. We can use these volumetric measurements or 3D measurements over time to assess changes in tumor size and therefore response to treatment. And so thinking back to our example of Jane. So again, here are the 4 MRIs that from her first round of chemotherapy treatment. And just to remind you, this 4th time point is the time point when the radiologist actually determined that the tumor had grown, even though she was on chemotherapy had already been on chemotherapy for 8.5 months. Now, what we did is we took our model and we said, okay, give us the 3D segmentation for each of these time points. So that's, you can see that in green here is the model predicted 3D segmentation. We did that for each time point separately and calculated the 3D size of the tumor across these different times. And this is these are the actual results for Jane. So these are the 4 time points here, these 4 dots. And we can see that not only was the tumor increasing in size consistently over time, but even within the first time, the difference between the first 2 time points, you can see an increase in the size of the tumor. And so just to relate this back to the timeline, this is where that model detected change or increase in size happened. This second arrow is when the radiologist had originally called it. And so that means that this model detected increase in tumor size, in the growth of the tumor, happened almost 6 months earlier than the radiologist. So that would have been 6 months less time on chemotherapy, 6 months less side effects, less long-term influences on her life, less time for the family to be sitting around waiting to see if this chemotherapy trial would work for her or not. And then if we start to expand this and think, okay, well, what about these other time points, again, where the radiologist was doing their best with these methods, but which still required a significantly long amount of time to determine that her tumor was slowly growing. If we think about this same difference in time at other time points in her treatment journey, that would have been 1.5 years eliminated of waiting around for based on the standard practices to detect tumor growth. And that's just major, right? This is essentially why we do what we do, is to be able to eventually have this exact type of impact for our patients. So clearly 3D has extreme promise to do a lot better than 2D. I'm not going to sit here and tell you that it's perfect, because it's not, but it has the promise to be to really impact patient care. So what we need to do is we need to iteratively make our AI model better, to be able to work across any patient that comes in at any time point in their treatment. Right now, what it does is the model performs really well on pre-treatment MRIs for the diagnostic cohorts, right? The cancer types that we have delineated manually. But we need to expand so that the model can see more and more time points to be able to work on, post-surgical MRIs, on treatment, during treatment time points, and just more subjects. So particularly for patients that are in those lower frequency diagnostic cohorts, we need more data in order for the model to perform well for them as well. We also are working on translating our model into clinical trials to test this longitudinally or over time. And ultimately, the goal is to implement this in clinical care, again, as is being done more and more, or is getting closer and closer in for adult gliomas, but just has not come to fruition yet. I'm going to talk briefly about our collaboration with PNOC, the Pacific Pediatric Neuro-Oncology Consortium. So what we're doing is we're partnering with them to generate these 3D segmentations for PNOC clinical trials. The goal here, again, being to standardize those measurements of tumor size, to perform the growth analysis using those volumetric growth curves, and to compare essentially the assessments of response that would happen with 3D with the actual 2D radiologist measurements. We additionally are using this as an opportunity to essentially create a database, including all of those volumetric measurements and all of those growth curves based on our 3D model, combined with all of the clinical trial information that's collected at the patient level. So all of that phenotypic, demographic, treatment response, adverse event data. And the goal really there to build out databases to enable synthetic and external control arm generation for future trials, ultimately to eliminate the need for patients to go on placebo arms. Okay. So that is one of our more advanced projects. And I'm going to talk now a little bit more about how we're using AI to, again, build out models to perform predictive analytics with the hope of translating back to clinical care. So bringing it back again to this MR image, right? So this is exactly what a radiologist would see at their workstation. This is essentially just, at this level, is just pixels. It's these images taken. There are different types of images within an exam. But at the end of the day, all you can do is describe qualitatively an impression based on just pixel level information. Our 3D segmentations allow a little bit higher level or further insight into the properties of the tumor. So you can think about it in terms of the volume, the shape, all of the different types of subregions that a specific tumor consists of. And the idea here is that we're trying to do is essentially see what types of patterns and whether there's really meaningful information that we can extract from the images beyond just the pixel level data. So that can be things like texture or 3D based modeling and really the combinations of all these properties together. So can we gather meaningful insight into the patterns in the images, and combined with other types of data, does that result in better predictive power? So we have several ongoing research projects essentially utilizing MRIs to see whether they have predictive power in clinically relevant tasks, and particularly when combined with other data types. Okay, so I'm going to start with a project again led by Dr. Fatih Kazaruni. This project looks at the prediction of patient survival. So essentially the prognosis of patients using MRIs and clinical data. And so essentially what's done here, again, we take CBTN data, imaging and clinical data like demographics or certain surgical data, like whether they had, residual tumor left over or not. And we feed all that data into, again, an AI model to try and learn patterns that will predict the survival of a given patient and group them into kind of high risk, medium risk, and low risk groups. So let's start with just the clinical data, right? So if we take just those demographics and other clinical information that we have about a patient, let's try and predict survival. So here what I'm showing you is a survival plot, essentially from left to right is time. The red line represents, patients who are at a higher risk of survival. So that's bad. These patients essentially just won't live as long as the other patients in this group. The blue is intermediate, and the green are patients that are expected to live longer. And those are the low risk. That's the low risk group. But what we can see is that there's a lot of overlap here. So these lines are not separated. So in other words, the model doesn't really do a good job at dividing patients into these 3 risk groups. If we feed a model just the properties derived from the imaging data, we get a little bit of separation here. So the model does a little bit better at separating out patients essentially who, again, are at higher risk or lower risk of survival over time. But really the amazing power that comes is when you start combining these data types. So the combination of imaging and clinical data together fed into an AI model does the best. It performs the best at separating patients into these risk groups separately, meaning that with a patient's MRIs and with their clinical data, we can have this model predict whether they're more likely to live longer or not. And based on that, the clinical implication here is to guide their treatment plan. So if a patient is at higher risk or lower risk, then we can tailor their treatment plan accordingly to be more or less aggressive. Another research project that we have essentially looks at correlating MRIs with tumor mutations. So based on the molecular data from the CBTN patients, So what the approach that was taken in this project was to essentially use the imaging properties to divide patients into, again, 3 separate groups here, but not really knowing what the significance of these groups were initially. Once we started looking at the molecular mutations of the patients in each of these groups, it was found that there were some mutations that were significantly different, meaning that there is a relationship between these imaging properties and the molecular status of the patients. Again, the clinical implication here would be to predict the mutational status of a patient using just MRI. So that really is the goal. Again, to provide therapies tailored to the patient. So if you think about Jane, she had a specific type of mutation of a subset of low-grade glioma, and those that information can be used to examine what types of chemotherapy, for example, the patient might receive, or whether they receive chemo and radiation. Okay, another research project is done. Yeah.
There's a question, if this holds true for high-grade glioma.
So right, this I'm assuming being the mutation study. So this was done on just low-grade glioma subjects, essentially because we had enough data in that cohort to be able to perform this analysis, but we're currently trying to expand to high-grade glioma patients as well. And so that's an active area of development, is to essentially try and get to build out these data sets so that we can exactly do those types of tests.
And then Kim had asked, does the predictability increase with the number of clinical data fields? Or some clinical data fields more useful than others?
Yeah, no, that's a great question. I mean, there's definitely differences. I think so I guess I'll go back to like this, for example. The idea is that, is there a minimal data set which is highly predictive? So more is not always better. Yeah, I think that's a great question. That's not necessarily something that we looked at in this data set. There are certainly, especially for some cancer types, there are certain attributes that are more relevant that have been shown to predict survival separately. Again, an awesome question. Okay.
There's a lot more slides to get.
Yeah, that's okay. I can, this is the good stuff. So let's see. So another research project that we're doing is combining our MRI data with pathology data. So it's just like radiologists kind of do in the clinic today. The pathologists have a similar workflow. So they'll take these slides of the tissue specimens collected in biopsy or a larger surgery, and they'll look at the slide under a microscope and they'll say, okay, here's my descriptive analysis. Here's, I see more density of tumor cells in this region and less over here. These are some irregularities or abnormalities. But really, again, that's a pixel level description of the image. So what we're doing, so Dr. Arya Matapfar is a neurosurgery resident at Jefferson Health. And what he's doing is essentially saying, okay, can we extract information similarly doing segmentation of these pathology images? Can we combine those with imaging to, again, provide better predictive methods of predicting molecular status, of predicting survival in certain cancer types? All right, and then the last research project is looking at, in a similar fashion, correlations between imaging and liquid biopsy metrics of our CBTN subjects. So liquid biopsies being taken through blood and CSF. We are trying to correlate imaging with these measurements to essentially, the goal ultimately would be to derive imaging properties that can predict liquid biopsy metrics to potentially provide non-invasive methods. So we don't necessarily have to have our pediatric patients undergoing like lumbar punctures, for example. All right, so I've talked about this so far. So Our goals here in the near future are to expand to more diagnostic cohorts, acquire more imaging, which I'll talk more about, and then also deal with this alignment of imaging with other data types and software tools, which is just an ongoing challenge. So really the wealth of CBTN, as many of you are aware already, but just to kind of underscore, is that we have this combined or this integrated data set. And that's really where novel characterizations and novel treatments can come out of, but it's still somewhat difficult to use this data in practice. So there currently, for example, is no software tool or workflow to do a really easy, streamlined integrative analysis. So this is another kind of unmet need moving forward. All right, the last part of my talk is essentially discussing all of these, all of the work that needed to be done in order to get to the point in our research projects where we are at. So really what we did to build out large data sets and processing workflows to make those research projects possible. So we at D3B are the data coordinating center for several multi-site consortia, like the major one being CBTN. This involves data storage and management, removing, identifying patient health information from the data files, and ultimately providing researchers access to those data sets and also technological resources so that they are able to use the data in their research projects. Some of the challenges that we faced initially with imaging data were that images are not like other data types. So images in particular are very heterogeneous in how they can be saved. There is a lot of information about the images that is required for use in research, but that some anonymization tools might wipe out. So there has to be really robust processes to be able to handle PHI, but also retain file information. And they're also unique file types. So they come as DICOM, what's called DICOM. At scale, CDs are just intractable. So a common practice is to use CDs to essentially ship images, MRIs, like for example, for referrals between hospitals. But when you start talking about doing this at scale, it's just not feasible. If you think about Jane having like 50 MRIs, for example, there's no way a team could handle that for the thousands of CBTN subjects. Images are large, and there was essentially no pediatric data set available. So there was no really model or infrastructure for us to use. So in brief, we essentially developed a software system to be able to do this and to do it robustly for CBTN. And what we do is we essentially take in the images, we process them, and then we put them on a website called Flywheel, where they can be processed further. We can run our automated 3D segmentation tool, and researchers can have long-term access to the data there. And they just are able to log in on a standard website and be able to download data and use it. To date, we've had 5 contributing institutions. We've collected, again, clinical MRIs over as many diagnostic cohorts as we could have include. So the data set is massive to date, but it's also significantly diverse. And as we start looking at our smaller frequency or diagnostic cohorts, like our ATRTs or our schwannomas, we still just don't have a sufficient amount of data for those groups to be able to do meaningful AI work with them. A lot of this data set comes from CHOP. And so really our goal over the next few years is to increase the contribution across sites. I'm going to just hop through a little bit. So we have this, we've done amazing work to bring in a large data set. What time is it?
We have 10 minutes, but I think
okay.
Really the background to that, I think that the real takeaway that I took when we were going through this earlier is that it's really, really hard to pull together all of these different images from different sites that use different machines that have different ways of annotating things. And that's taken a while to figure out. And that's why nobody else has done it before.
Right, exactly. Yeah.
So
No,
that's what all of those
100%.
slides that
Yes.
we have through,
Thank
that's
you
what they
so
were
much,
saying.
yeah.
Yeah.
Right. Then there were additional kind of processes we had to build into our software systems so that we could also use that data. So clinically acquired data is messy. Exactly like Jerry said, we needed to annotate it, and we can't really expect each research team to sit there and comb through these thousands of files that we're providing to them. So we needed methods of user-friendly data exploration and data preparation. Imaging also in particular has a lot of manual steps required to prepare the images for AI use. And that often requires a lot of technical expertise, and it's difficult to perform and to do QC at scale. So just to show you a little bit about like the variability in clinical practice. So each of these columns is one type of MR image, and each of these rows is what that image could be called at even just within a hospital, but particularly across hospitals. So a researcher might be only interested in kind of seeing, okay, I want all images of this one type, but they would have to sit there and comb through all of the images that are called all these different things in order to select those. And so what we did is we use essentially a large scale labeling process so that we build out meaningful labels and annotations of the data files to be able to allow researchers to perform data exploration. We database that, we provide a website that's publicly available for people to go and explore our data set, and also explore and filter based on other clinical and molecular CBTN data types. But nonetheless, a human in the loop is still required. So we're still just continuously improving and iterating on this process to make it feasible and to make it scalable. All right, so bringing it back. So back to the overarching goals. So again, our aim is to support this end-to-end life cycle, right? So as data comes in, it easily gets fed into research contexts. Those research models that are developed can be easily and consistently applied to clinical data so that ultimately the models can be used to actually guide patient care. So we really want to support this entire life cycle using uniform ecosystems and software tools. Researchers should not have to worry about combing through files at the end of the day. They should be able to just take the images that they need and focus on making discoveries with them. And so some of the ways we do that I've touched on are through these open data sharing practices and through rapid data preparation and tool generation. Like I said at the beginning, our goal really is to enhance and improve patient care. We do that through building out AI-powered software tools, scalable image processing pipelines, and standardized workflows of implementing these AI models. So really, like, the dream in the future would be that Jane would have come in on day 0. She would have had a clinical MRI collected, and if she did have a brain tumor, then a report would have been generated based on those MRIs, giving as much characteristic information as possible. So accurate measurement of size and accurate characterization of her genetic mutation and other types of factors and properties that could have better guided the treatment that she ended up getting. Okay, so this is essentially where we're at now. So just to kind of bring it back with where we're going in the near future. We have this automated tumor segmentation model. We know that it can be used pretty robustly for patients at before treatment time points. And we have built out an infrastructure to support the processing of these MRIs at scale. We are, again, aiming to build out into clinical trials so that we can ultimately use this model across clinical contexts, across patients from different disease types, and so that it can be used in clinical care, but it needs more training. We want to expand to other exam time points and more cancer types and test the model in clinical trials. And so really where we're going is we want to essentially grow our team in the next couple of years. We need more imaging data analysts, data scientists, and really the goal there being that we could help sites with their bottlenecks in data collection. So we could collect, for example, all retrospective CBT and MRIs and set up prospective transfer workflows. So clinical research coordinators wouldn't have to worry about downloading and uploading MRIs, which just is super laborious and time consuming. But that they could, at a click of a button, be able to send all MRIs for current patients. We also are focused on developing out data processing workflows, again, to make this easier, to make the MRIs easier to acquire at large scales and to prepare it. And really, if we were able to do that, the success would be defined as eliminating what current human resources are needed to, again, acquire this data and prepare it for building out AI models, because it just takes so long and it's very manual. So really decreasing the cost of coordinators, doing this manual work, increasing their efficiency, and ultimately allowing them to focus on areas that will directly benefit the patient as opposed to downloading MRIs from their radiology department. Ultimately, we want to grow out the CBT and imaging data set so that we represent all cancer types, and that we can build AI models that will perform well across all of them. All right, and with that, I have no clue what time it is.
We have 3 minutes.
Oh, sweet, I made it.
So I have a question here. Will this AI model be disease agnostic, or will it will it be for specific?
Yeah, that's a great question. So the goal is for it to become that. Like has been shown, right, with other data types in CBTN, the strength really comes in multi-site collaboration and multi-site data aggregation, because for some of our lower frequency cohorts, data from a single hospital just isn't enough to be representative. We need to aggregate across hospitals. And so we're trying to do that. Unfortunately, a lot of our imaging data comes from CHOP because we've been able to kind of like get our hands dirty and really build out these automation pipelines at our institution. But the goal is to expand to be disease agnostic.
Yeah, so that, I mean, that's the power of CBTN overall, right? That we're trying to collect all and aggregate all of this data to inform in clinical decision making, and also to inform discovery. So that's what CBTN is all about, and we're just trying to bring the imaging piece up to speed with the clinical data and pairing that with all of the molecular data that we've been working towards. Oh, what about international patients?
Yeah, definitely. Yes, that is the goal. The goal is for anyone to be able to go to our Flywheel, for example, our website, or a different tool even, where we can input our model and have it loaded there so that anyone could take their MRIs and go in and essentially plug them in, and it would work and generate an accurate 3D segmentation.
Oh, and then, Kim asked this general question, perhaps from application of AI, machine learning in other areas, is there a threshold of how much data is needed
before the predictive value becomes extremely accurate? Yeah, no, that's a great question. Essentially, it depends. But yeah, I mean, greater than 90% is a wonderful kind of not really statistically justified by any means, but is definitely a kind of like a high confidence threshold. So for example, if you're thinking about the 3D segmentation work, or even other types of predictive models, performance is thought to be good above 80%, but ideally if it would be clinically relevant, it would be much higher, much closer to 100%. In other domains, I think they might, care a little bit less about accuracy. So when it comes to self-driving cars, you'd want to be closer to 100%, but for sales and advertising, they care mostly about just it's less relevant for the outcome if it's a little bit lower accuracy.
I think we covered, are there any questions that we didn't cover with people still on after we're a minute over? Well, I'm so glad and just delighted that we got to share this awesome information with all of you. It's really exciting to see to see the progress that's been made. And now as I talk to so many of you and I continue to gush about it, you'll have further background about everything that we're trying to do here. So really, thank you again for joining us. Ariana, thank you so much.
Of course, thank you so much.
We recorded this, so we'll share it out with everyone. But if you have other questions, please don't hesitate to reach out to us. We're happy to I'm happy to pester Ariana on your behalf.
Thank you all so much for your participation and for having me.
Yes. All right. Have a great day, everybody. And I'll shoot out a note with the recording in probably before the end of the week.